Hello, my dear students. I hope you all have been doing well. And from today, we are going to start another part of our journal science class, that is the biochemistry part. And in this second part of journal science class, we are going to discuss about the basic concept of biochemistry. And this part would be also consisting on 2-3 lectures, and we are going to discuss in this part about different molecules, biomolecules. We are going to discuss about protein, we are going to discuss about lipid, and we are going to discuss about amino acid, carbohydrate, and we are going to discuss about their structures, their metabolism, and their physiochemical properties. So in this lecture, that is my technically this is my lecture number 4 of journal science class, but from the part of basic concept of biochemistry, this is my first lecture. So to understand this lecture, we have to understand about the atoms, and what are elements, what are atoms, that we have already covered in our past 3 lectures. That were the part of my that were the part of the that were the part of the chemistry portion of the journal science class. So so we are going to start about that. What are any amino acid? First we would be discussing, and then we will be going to discuss about more advanced concept, that how amino acid join together and form the protein. So first of all, we have to understand, to understand protein, we have to understand that what are amino acid. And what are amino acid? Amino acid are biological macromolecules and biological molecules or biomolecules. And amino acid, as the structure we are seeing, actually this is the collection of atoms. And our previous lecture, we have already understand that what are atoms, and we have already discussed that what is the structure of atom. Now this is the beauty of science, that in science everything is start from scratch. So we have seen the atom, now we are going to see that how atom join together and make a big molecule. So these are the biomolecule. Amino acid are biomolecule. Why we say them as a biomolecule? Because they are having some biological properties. So that is why we say them as a biomolecule. So we are having a structure of amino acid here. When we look on the structure of amino acid, we are seeing here that amino acid is having one central carbon atom, that normally we designate it as a C alpha atom. And this carbon, normally if we talk about the chemistry of carbon atom, so carbon atom has capability to make 4 bonds. Why? Because it is having 4 lone pairs of electron in its outer shell, so they can pair up with some other electron that are coming from other atom. So always wherever we are finding carbon is having capability to make 4 means bond with other 4 bonds with other 4 atoms, or pair with some 4 others electrons. So in the amino acid, we are also having carbon atom, that is a central carbon atom. So it means it can be making 4 bond with different other atom. So in the amino acid, we can see this carbon atom that is having 4 different that is having ability to make 4 bonds. So in the amino acid, it make bond with one of the carboxylic group, one of the carbon atom of the other carboxylic group, and one with the one of the nitrogen atom of the amino group, and one of the atom that is coming from the R group. So what is this R group? It is also again the collection, we can understand like this. This is R group, is also collection of the atom, or it is another molecule, chemical molecule. So when I talk about the structure of amino acid, how amino acid looking like, we can just remember that amino acid is a biomolecule that is having a central carbon atom, and this carbon atom having capability to join four other atoms. So I forgot to tell you that the last means bond that central carbon atom is making, it will make with the hydrogen atom. So amino acid is a biomolecule that is having a central carbon atom, and this central carbon atom is found to join with one carboxylic group, one amino group, one hydrogen atom, and one of the R group. And this R group is found to be different in different type of amino acid. So different amino acid are unique because of the differences in this R group. So here we understand completely that what is an amino acid, and how its structure look like, and what is present in this structure. So once anyone will be asking you that how you define the structure of amino acid, you just have to say like that amino acid is a biomolecule that is having a central carbon atom that is found to join with one carboxylic group, one amino group, and one R group, and one hydrogen atom. So these amino acids join together and make a long chain that we say as a polypeptide chain. Is the polypeptide chain then further fold on itself and making the protein. So actually the protein is a macromolecule, and amino acid is a biomolecule, protein is a macromolecule. So what is the difference between a biomolecule or a macromolecule? Difference is that a simple molecule is like the amino acid. It is a just single entity. So what is a macromolecule? Once many biomolecules join together, they make a big molecule that we say is a macromolecule. So protein is a macromolecule, and protein is also means act like a biomolecule because it is also having biological activity in our body. So you have to just understand here that amino acid when joined together, they make a protein molecule. And in nature or in our body, we can find 20 or sometimes 21 amino acid. And here we can see that what are amino acid with the different color dot. It is showing that different type of amino acid. Once they join with a bond, with that, this name of this bond is a polypeptide peptide bond. So once these amino acids join together with a peptide bond, they make a long chain. We say it's as a polypeptide chain, and then we can see here this polypeptide chain fold itself and make a protein. So this is the thing of there. And now we are going to discuss, we would be we would be discussing in detail more about the protein in my next lecture. But first we are going to study here that how many means amino acid is existing in nature, and how their structure looking like. So I have already told you that we are having 20 or sometimes 21 amino acid in our body, and these amino acid are having different means structure. Each amino acid having its unique structure, and because of the difference of the structure of the R chain. But you have to remember that each amino acid would be having, if I'm going to back, each amino acid would be must having one amino group, one carboxylic group, and one hydrogen atom. All the amino acids that are existing in nature, they would be having three same thing, including the C alpha central carbon. And what these three group would be same in each amino acid, that is amino group, carboxylic group, and hydrogen atom. These three group would be same in all the amino acid, but all the amino acid would be having different R group, and all the amino acid are having different structure from other because of the different having different R group. So we can see here the red color. All these are the R groups, and this column is showing the structure of amino acid. Here we can find all the name of the amino acid, and here we are having this symbol. How we can use normally, what symbol we use normally in our textbook. For example, for the glycine, we can write gly, or we can just write G. And these letters are important when we are writing the sequence of amino acid. We would be understanding in the coming lectures why these letters are important. So you have to, this is very important for you to remember that which amino acid is designated with which letter. So glycine we designate with the G, and it is we short form of glycine is gly, and alanine Ella, and A, and valine val, V, and so on. And once we are writing the sequence of amino acid, we can also write gly, Ella, val, liu, or we also use these letters G, A, V, L. So that is why these symbols are important, and I am discussing here. So now I am going to the structure of amino acid. So we are going to start the understanding the structure of different amino acid from very basic amino acid, very simple amino acid, that is glycine. So glycine is one of the very simple, very basic amino acid that is existing in this earth. Why? If we are going to see the structure of amino acid, that is a glycine, that is very simple. We can see that although this amino acid is having one carboxylic group, one amino group, and one hydrogen atom that is attaching with the central carbon, but it is also having an R group, and in case of the glycine, the R group is a hydrogen atom. I am sorry, I told you that this red one is R chain. This red one is not R chain. This red one are indicating carboxyl group, amino group, central carbon, and hydrogen atom. So all these are red. That is showing that all these means three groups are similar in all amino acid. So the black one is the R chain, that is showing different in each amino acid. So in case of glycine, that is very simple amino acid, the R chain is hydrogen atom. So that is why the glycine is very simple and very basic amino acid. Why? Because its R chain is very simple, only single hydrogen atom that is attached to the central carbon atom. So now if we are going to see the means the structure of the alanine, we can see that in the alanine, although we are having same all three group present as they are present in the glycine, but in case of alanine we can see a different R chain that is a little bit complex than the hydrogen atom. And then if we go to the valine, we can see that all the three group, carboxylic, amino group, central carbon, and the hydrogen atom is same as the previous two, and we are having a more complex R chain. So as we go down, means as I am going forward, I can see this R chain is getting complex in other amino acid. So due to the differences in the R chain, amino acid is having different physical and chemical properties, and their uniqueness, each amino acid is unique from other amino acid and having different properties because of this unique R chain. So we are having cysteine, and we are having methionine here, and we are having serine also, and we are having also threonine. You can see here, and then we are having tyrosine that we for the tyrosine we use symbol Y. So it is important. We can see here, if I am going up, that it is very easy to understand that glycine we use G, and up till now for leucine, isoleucine we use I, and methionine we use M, and for cysteine we use S, It should be C. Okay, and methionine we use M, but for serine we use S, but for threonine we use T, but for the tyrosine we use Y. So we get means confused with the tyrosine. For tyrosine we do not use T, we use Y. So you have to understand, means keep this in your mind, that for the tyrosine we use Y. And for aspartate, although it is starting from A, aspartic acid, it is starting from A, but we do not use symbol A for that, we use D. So you have to means memorize it, that for the aspartic acid we use D, for the asparagine we use N, for the glutamic acid we use E instead of G, and for glutamine we use Q instead of G. And we can see here all that again we are getting R chain that is getting complex. And for the lysine we use K, and short form of lysine, abbreviation of lysine is Li. And for arginine, ARG, we use R, and for histidine we use H. And we can see all the amino acid having different chains. I'm going to discuss with you different amino acids separately, but here we are just seeing that how we can abbreviate amino acid and what is looking different here, and how difficult it would be for us if we are going to write the abbreviation of these amino acids. For phenylalanine we use F instead of P, and for tyrosine we use Y, and for tryptophan we use W instead of T. And we can see that we are having different R chain but same amino group, carboxylic group, hydrogen atom and central carbon atom. And the last amino acid, that is the proline, we use P, and we can use short form as pro, and we can see its structure here. So these were all 20 amino acids that are existing in this in the nature. And now we are going to discuss about the how we classify these amino acids. So we are having several different classification. So two most important classification that we use to classify or to put together different type of amino acid in different groups. So one is based on its chemical properties, and the other is based on the nutritional requirement. So I'm going to discuss about first about the all those amino acid, what is the classification that we use on the basis of the nutritional requirement. So nutritional requirement, we classify amino acid into three group. One is the essential amino acid, one is the semi-essential amino acid, and the other one is non-essential amino acid. So the essential amino acid are all those that we absolutely need in our diet. They are not synthesized by our body, and we need to eat them, we have to take in them in our body from outside of the source, from the food. So what all these are amino acids? These are isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. And the semi-essential are those that can be synthesized in our body but not in adequate quantities, and we need to have some of their portion from outside of our body or from our diet. And non-essential amino acid are those that are not absolutely we are not absolutely need to take them from the outside of the source or from our food, although it can be present in the food, but they are already synthesized in our body, and we are okay with them. So these are alanine, asparagine, tyrosine, aspartic acid, cysteine, glutamic acid, glycine, proline, serine, hydroxylysine. This is, I think, modified amino acid, glutamine, and hydroxyproline. So this hydroxylysine and hydroxyproline, these are some modified amino acid. And if I am talking about the classification of amino acid, that is based on its chemical properties. So I am having non-polar amino acid, polar amino acid, aromatic, positively charged, negatively charged. So first we are going to see that what amino acid are in place as a polar, non-polar, aromatic, positively charged and negatively charged. Then we are going to extensively review them on the basis of their structure, and we would be seeing that why we say them polar or non-polar. So for the non-polar amino acid, we are having glycine, alanine, valine, leucine, isoleucine, methionine, and for the polar, we are having serine, threonine, cysteine, proline, asparagine, glutamine. For the aromatic, we are having three that are very most popular amino acid, and wherever it is coming the name that aromatic amino acid, you should have to just quickly say that it is phenylalanine, tyrosine, and tryptophan. And for positively charged, we are having lysine, arginine, histidine, and for negatively charged, we are having only two amino acids that are in this nature that are present as a negatively charged amino acid, that is aspartic acid and glutamate. And now we are going to see why we are saying them that they are polar or non-polar. So if I'm saying our first classification is all those amino acids that are non-polar in nature and aliphatic, having aliphatic R group. So first we have to understand what is this aliphatic. So once we are having ring structure, so this is aromatic thing, okay. But once we are having a open chain, like we can see here, open chain, there is no any ring, so we say it's as a aliphatic. So these the highlighted things, these are the R group, and as we see that this is a central carbon, this is amino group, amine group or amino group, and protonated amine group, protonated amino group, and then it is carboxylic group, hydrogen atom, and this is the R. So this is the R. So we see all these three things, three groups are same in each amino acid. And the highlighted one, these are R group that are different in each amino acid. So what are the aliphatic R group? If we are having R group that are having present as an open chain, not present as a very closely ring, so this would be aliphatic R group. We are going to see also the aromatic R group. So let's see, then you would be understand then what is the difference between aliphatic and aromatic. So now you have to understand right now that what is the aliphatic. Aliphatic is the open chain, and what is non-polar, that is not having any polarity. So what is a polarity? Polarity is the capability of any molecule to make a bond with the water or to make association with the water. So any molecule would make the association with the water, means able to attract the water or other solvents, polar solvent, on that time when that molecule would be having any charge, either the positive or negative. So if a molecule is having any ability to attract any other molecule on the basis of its charge, we will be saying that it is a polar, okay. But what is a non-polar? Non-polar is that is not having capability to attract any other molecule. Why it is not having? Because it is, although maybe it is not having any charge, or they are having any charge, but charge are cancel each other. So all overall charge on the molecule would be zero. So as we are having in the case of these all amino acid that we are having here, for example, first I am going to talk about the glycine. So why glycine is non-polar although it is having positive charge and negative charge? Because glycine is having only one positive charge and one negative charge. So one positive and one negative is cancelling in each other, and overall charge on this glycine would be zero, okay. So that is why it is the non-polar amino acid, but still it will be having so it will be non-polar, it is not having any polarity. So if I'm talking about the alanine, we can also see the alanine is having one positive charge on its amine group, and one negative charge that is present on COO, and these two positive and negative are cancelling in each other, and overall charge of the alanine would be zero. So this is how these all amino acids are non-polar, because although they are having charge, but the charge are present in the equal and uniform quantity, and they are cancelling with each other. So all these amino acids, glycine, valine, proline, Glycine, alanine, proline, valine, leucine, isoleucine, and methionine are all these amino acids are non-polar, and these are having aliphatic R chain, means they are having open chain. So another type of amino acid, another classification of amino acid is the aromatic amino acid, or all those amino acids that are having aromatic R chain. So here we can see that all the amino acid having same amino group, carboxylic group, hydrogen atom and carbon atom, but these highlighted R chain are different. Okay, and we can see in the form in here we can see here that these R chain are having this ring structure. So this is a closed ring, and we say them as a aromatic. Why we say them aromatic? Mostly it is a benzene ring. And why we say this aromatic? Because this ring is having conjugated double bond. What is a conjugated double bond? A double bond that is present alternatively position in the benzene ring. Okay, so this is having conjugated double bond that is present in alternative position in the benzene ring and in the ring. And these are but you have to understand that aromatic amino acid having closed ring structure, and this double bond, conjugated double bond, give this ring as a aromatic behavior. So we absolutely do not need to study about aromaticity right now. But just from this picture you have to understand that we are having aromatic amino acid, who all those amino acid are aromatic amino acid. Typically there are only 3 amino acid that are existing in nature that are aromatic amino acid, and these are phenylalanine, tyrosine and tryptophan. So these amino acid are having a closed ring R chain, an R chain that is having closed ring. So this is how that these aromatic amino acid are present. And then we are having another classification, that is that are those amino acids that are having polar in nature, and they are, although these are polar, but they are not having any charge. They are having R group that is uncharged. So we are having two things here, that we are having amino acid that are having R group, but R group is uncharged, but still these amino acids are polar. So we can see here why these are polar, although these are uncharged R group. So we can see here we are having serine, threonine, cysteine, asparagine and glutamine. So we can see all these 3 things are present, same would be same amino group, carboxylic group, hydrogen atom in all amino acid, and then highlighted R group would be changed. So we can see that in the R group we are not having any charge here, but these and we are not having any charge, we are having hydroxyl group, and here we are having one charge, positive charge, and one negative charge. So these 2 charges would be cancel each other. So overall charge of this amino acid would be 0, but it is polar. But why it is polar? Because of the presence of this hydroxyl group. So this hydroxyl group is by default, it is having ability to attract other molecule or water or other solvent. So if this hydroxyl present in any other molecule, so it would be polar. So here in serine we are having hydroxyl, so this amino acid would be polar because of the presence of this hydroxyl in its R chain. So now second we are going to discuss about threonine. In the threonine we are going to have again hydroxyl, so it would be polar because of the presence of this hydroxyl. And in the cysteine we are having SH, so this amino acid would be polar because of this cysteine SH group. And then we are having asparagine, it would be polar because of this group. And then glutamine, it would be again polar because of this group. So these are the polar, and all the amino acid would not having any charge, but they are having polarity because of the presence of any group in their R chain that would be having polarity. Now we are having positively charged amino acid. So we are having 3 positively charged amino acid in the nature. One is lysine, one is arginine, and one is histidine. And we can again see that all three amino acid are having 3 same group, amino carboxylic hydrogen, and the difference in the highlighted portion, that is the R group. And now we are going to see why these are positively charged. So we can see that these amino acid having a group that is having a positive charge, these amino acid having R chain that is having a positively charged group. Okay, and then what will happen, we are already having a positive group here, a negative group, positive charge here, a negative charge here. So what happened, this positive and this negative would be cancelling in each other, and it will be leaving this positive charge remaining on this overall molecule. So that is how this overall molecule or amino acid would be having a positive charge, or we say them a positively charged amino acid. So same thing would be happen there, this positive and negative cancel with each other, and this R group would be having only one positive charge. So because of this positive charge, this overall amino acid would be having positive charge on it. So same thing would be we would be having here. So these are positively charged amino acid. Now we are going to discuss about the negatively charged amino acids. As from the name it is indicating that these amino acid must be having a negative charge on it. So we are going to see how they are having negative charge on it. We are having 2 negatively charged amino acid, one is aspartate, one is glutamate. And if I am talking about the aspartate, we can see that we are having one positive charge, one negative charge, that is going to cancel each other. And in R chain we are having a COO negative group. This is a group that is present in R chain. Because of this group would be giving overall charge on this amino acid. So these are the negatively charged amino acid. And then it is another is a glutamate, and it is it would be having same negatively charged. Why? This positive negative cancel each other, and then this amino acid would be having overall negative charge because of this COO minus. So these were all different type of amino acid, and we see that how their structure is looking like, and why they are positively charged, why they are negatively charged, why they are polar. Now we are going to discuss about the physical and chemical properties of amino acid. So actually this structure and this R group in the amino acid give them ability to behave differently in the chemical environment or in the in simple environment. So for example, if I am talking about the aromatic amino acid, so we are having 3 aromatic amino acid: phenylalanine, tryptophan, and tyrosine. So these amino acid are having ring R chain, ring structure in their R chain, or ring group in their R chain. So for the phenylalanine, this group is a toluene, and for this ring is a toluene. And this for the tryptophan, we say this ring as an indole ring, and then it is a closed ring that is having conjugated double bond. And for the tyrosine, we are again having toluene that is attached with a hydroxyl, and this ring is having conjugated double bond. So because of the presence of this closed ring that is having conjugated double bond, these 3 amino acid having capability to absorb light, UV light. Okay, so this is a very important properties of amino acid, and we can even detect protein because of this ability of absorbing light by these amino acid. For example, if we are having a mixture of amino acid in a glass or in a test tube, and we do not know that we are having a mixture of protein in a test tube, and we do not know what type of protein are present in a test tube, so we can keep it under UV light. And if all those protein type of protein that are having absorbing a lot of light, means UV light, in UV region. So we can say them this protein is having a lot of aromatic amino acids. So different protein are having different type because of the having different type of amino acid in them. So this property enable us to detect different protein or to identify the protein in a group of unknown protein, or to classify the protein also. So this is one important property of the amino acid, and only we are having 3 amino acid that could be able to absorb the UV light. That is the tryptophan, phenylalanine, and tyrosine. Now another chemical property of amino acid is chirality, or we can say is, I think so, chiral, but I say it's a chiral center. So what is it? Chiral is a when an atom, a molecule having a central carbon atom, and this central carbon atom would be connected to four different carbon atom, four different other atoms, so it would be achiral. But if we are having a molecule that is having a central carbon atom, and this central carbon atom is not connected with four different other atom, so it would be chiral. Not chiral. A is not chiral. If a central carbon atom would be attaching with different, so it would be chiral center, but it would be attaching with the same, it would be not chiral. So this chirality is showing chirality is linking with the different group attaching with the central carbon atom. So because of this chirality we are having some stereoisomers. And what is a stereoisomer? Stereoisomerism. What is the stereoisomerism? Stereoisomerism is the means arrangement of the atom in a molecule around a central carbon atom. So we say it's a isomerism. What is a isomerism? Once different atoms are arranged in a space around a central atom by different ways, so we say it's a isomerism. So because of this chirality, amino acid is having two stereoisomer or two spatial arrangement. So one is the left-handed amino acid and one is the right-handed. And we can see here, for example, I am having an example of alanine here. So this group is on the left side, and if it is on the left side, we say it's a L-alanine. But if the same group is present on the right side, so it will be D-alanine. And this amino acid would be having different isomerism or isomer because of the having of the chiral center. Because of the chiral center means because of the attachment of the different group around the central carbon atom. So amino acid that are having different two means isomers, that are also enantiomers. And what are enantiomer? Enantiomer are all those amino acid that are mirror images of each other, but they are non-superimposable. So another important property of amino acid is an optical activity. Because of this ability of amino acid, because of the ability of amino acid to have a different means atom joined with a central carbon atom, or different groups joined with a central carbon atom, or because of the ability of the isomerism, so amino acid can be rotated in a plane polarized light, either left hand side or the right hand side. So all those amino acid, we say this activity as a optical activity. Once the amino acid can be rotate itself when we keep them under the plain polarized light. And the other chemical properties of amino acid is they can be you can find them as a dipolar ion on a physiological pH. So what does it mean by dipolar ion? You can find them as a dipole ion or in a physiological pH, and dipole ion we can say them as deuterion. So amino acid act as a deuterion on physiological pH. So what does mean by deuterion? Deuterion is an ion that are having both charges, positive and negative. So amino acid is having this capability or this ability. They can be act as a deuterions, means they are having two ions at the same time on a physiological pH, or they are having two poles. If they are having two charges, so these charges create the pole on them. So amino acid can be act as a deuterion on physiological pH. So because of this deuterion, amino acid can be exhibiting acid-base properties. So means amino acid can be act as an acid or a base, or as a base. So here we are having an example of amino acid that are positively charged amino acid, and we are going to see how that amino acid can be act as a acid or it can be become a deuterion. So let's see, we are starting from here. First we are having a positive charge amino acid, and the amino acid act as an acid base on a physiological pH. Why? Because they can gain proton or they can lose proton with the change of the pH of the body fluid, or if we are going to keep them in the test tube and we are going to change the pH of the solution, so amino acid can be acting as a base or an acid. So how it will be happening like this in our body in a test tube, we are going to see there. For example, going to talk about first with the positively charged amino acid, that are basic amino acid. We can see this amino acid is having a positive charge. So once at means neutral pH, it would be present only positive charge, but once the pH is going to increase, means so amino acid would lose its proton from carboxylic group, and then it would be having both charge. Okay, that one is positive and one is negative, and this would happen at nearly means acidic pH. So what will happen, this amino acid, now we are saying it's a conjugated base, and we would be saying it's as a deuterion that is having two charges on a nearly physiological pH, not exactly the physiological but near the physiological pH. So as the pH will be going to the basic. So what happened, then it will again lose one proton from its amino group, and then it will become the acid. So this amino acid start from the base and then it end up with the acidic amino acid. So this is how the amino acid act as an acid and base on a physiological pH. And what is a PI? PI is a isosteric point, and PI is that pH in that amino acid would be formed mostly as a deuterion. Okay, so this pH, for example, 5.97 PI, we will say is a PI. PI is a isoelectric point. So what is a isoelectric point? Isoelectric point is that point on that we can find amino acid as a deuterionic form. So in a deuterionic form, the amino acid would be having, although having both charge, but it will be having no net charge, and at this point at the PI isosteric point, amino acid would be having minimum solubility, and mostly protein that would be having deuterion on, and we are placing them in a pH so in a solution that is having nearly pH 5.9 or near isoelectric point, so they would be precipitate. So this is the importance of the isoelectric point, and we can calculate up isoelectric point of an amino acid by combining the pKa1 and pKa2 and dividing them by 2. So it is very easy. And now we have discussed all this thing. Now we are going to discuss how amino acid is metabolized. So we can see that we can see the structure of amino acid, that amino acid having a carboxylic group, amino group, and a hydrogen atom and a R chain. So what happened if we eat these amino acid and we are taking from outside of the body, or if they are present in our body and we need to utilize them to generate the energy, so they have to metabolize. So how they are metabolized, we are going to see them. And so how they are metabolized, for example, we are talking about the glutamine. So glutamine is an amino acid. So for its metabolism, it will be enter from the extrahepatic tissues into the mitochondrial matrix, and by the enzyme glutaminase, it would be convert into it would be glutamine, it would be converted into glutamate. So yeah, and then glutamate, we would be having ammonia, and then bicarbonate would be adding in this glutamate, ammonia, bicarbonate, and this glutamate all together would be going to make carbamyl phosphate. And the enzyme that would be catalyze this reaction would be carbamyl phosphate synthase 1, and it would be releasing it would be using 2 ATP, and yeah, so this carbamyl phosphate then releasing one phosphate, and then it would convert into citrulline, and then citrulline would be enter in the urea cycle. So in urea cycle, what happened, this citrulline would be using ATP, 2 ATP, and releasing PPI, and it would become convert into citrulyl AMP intermediate. This is an intermediate, and then what happened, it would be using another amino acid that are aspartate. This aspartate and this citrulyl AMP intermediate would be combining together and would be making arginosuccinate. So this arginosuccinate then would be split into arginine and fumarate, and then this arginine then releases the urea and making the ornithine. So this urea would be excrete out from our body, and this is the complete metabolism, catabolism of the amino acid, that is glutamine. Once urea form in our body and excrete out from our body, so it means this amino acid, that is, we are having an example glutamine, so it means we have excrete out completely excrete out urea, and whatever ATP or energy generated during this process we have utilized up in our normal daily life, or to doing many biochemical reaction in our body. So this is how the amino acid metabolize in our body, catabolize particularly. So metabolism is including both anabolism and catabolism. So this is how these amino acid get catabolized in our body. And one thing more, that for example we can see that there is another molecule, that is oxaloacetate, by the enzyme aspartate aminotransferase, it convert into aspartate, and then this aspartate also utilizing here to make the arginosuccinate. It would be combining with this citrunyl AMP intermediate, and then would be making arginosuccinate. And then here we can see another thing, that glutamate once enter in the mitochondrial matrix. So with the action of enzyme glutamate dehydrogenase, it can make this ammonium, and then ammonium would be combining with the bicarbonate ATP and making carbamyl phosphate. So to make the carbamyl phosphate we need ammonium, and bicarbonate and ATP. So this is how then this carbamyl phosphate is a main thing that is convert into citrulline, and then citrulline is enter in the urea cycle and generate the urea. So this is how the amino acid catabolism is taking place in our body. So whatever amino acid we take from outside or that synthesize in our body, we utilize them, we catabolize them to generate the energy. And a lot of amino acid that we synthesize in our body, so they synthesize via different other cycles, and we say them as a anabolism. So up till now, thank you very much for your kind concentration to learn from me. And in the next lecture we are going to discuss about that how these amino acids join together and make the protein. So thank you very much and take care. Bye.